Greetings from Everyday GMP. I am your presenter from Everyday GMP. This is series of video. We will discuss all the definitions and term used in EU guidelines for good manufacturing practice for medicinal products for human and veterinary use. In this video we will cover terms starts from alphabet C. Further series will be covered in other videos series of definitions. Video number 2, link of first video covering A and B is in description. Keep watching all videos, so let's start. In the world of pharmaceuticals, precision and adherence to regulations are paramount. Understanding the key definitions and terminology laid out in the European Union Good Manufacturing Practice Guidelines, Annex 1, is not just important, it's essential. Join us on a journey through the intricacies of pharmaceutical manufacturing as we dive deep into the terminology that shapes this industry. In this comprehensive video series, we will demystify the definitions used in the EU GMP Guidelines Annex 1, breaking down complex concepts into easily digestible explanations. Exploring pharmaceutical definitions from EU GMP Guidelines Annex 1, unlock the language of pharmaceuticals and dive into the core terminology that shapes the industry. Join us on this educational journey to understand the key definitions that ensure precision and quality in pharmaceutical manufacturing. Subscribe and let's begin. So before start, subscribe the channel and press the bell icon. So let's start. Campaign manufacture, a manufacture of a series of batches of the same product in sequence in a given period of time with strict adherence to established and validated control measures. Classified area, an area that contains a number of clean rooms. See clean room definition. Cleaning, a process for removing contamination, e.g. product residues or disinfectant residues. Clean area, an area with defined particle and microbiological cleanliness standards, usually containing a number of joint clean rooms. Clean room, a room designed, maintained, and controlled to prevent particle and microbial contamination of drug products. Such a room is assigned and reproducibly meets an appropriate air cleanliness level. Clean room classification, a method of assessing the level of air cleanliness against a specification for a clean room or clean air equipment by measuring the total particle concentration. Clean room qualification, a method of assessing the level of compliance of a classified clean room or clean air equipment with its intended use. Closed system, a system in which the product is not exposed to the surrounding environment. For example, this can be achieved by the use of bulk product holders, such as tanks or bags, that are connected to each other by pipes or tubes as a system, and where used for sterile products, the full system is sterilized after the connections are made. Examples of these can be, but are not limited to, large-scale reusable systems, such as those seen in active substance manufacturing, or disposable bag and manifold systems, such as those seen in the manufacture of biological products. Closed systems are not opened until the conclusion of an operation. The use of the term closed systems in this annex does not refer to systems such as RABS or isolator systems. Colony forming unit, CFU, a microbiological term that describes a single detectable colony that originates from one or more microorganisms. Colony forming units are typically expressed as CFU per milliliter for liquid samples, CFU per M3 for air sample and CFU per sample for samples captured on solid medium such as settle or contact plates. Contamination: the undesired introduction of impurities of a microbiological nature, quantity and type of microorganisms, pyrogen, or of foreign particle matter, into or onto a raw material, intermediate, active substance or drug product during production, sampling, packaging or repackaging, storage or transport with the potential to adversely impact product quality. Contamination Control Strategy (CCS), a planned set of controls for microorganisms, endotoxin, pyrogen and particles, derived from current product and process understanding that assures process performance and product quality. The controls can include parameters and attributes related to active substance, excipient and drug product materials and components, facility and equipment operating conditions, in-process controls, finished product specifications, and the associated methods and frequency of monitoring and control. Corrective intervention: an intervention that is performed to correct or adjust an aseptic process during its execution. These may not occur at a set frequency in the routine aseptic process. Examples include such as clearing component jams, stopping leaks, adjusting sensors, and replacing equipment components. Corrective intervention: an intervention that is performed to correct or adjust an aseptic process during its execution. These may not occur at a set frequency in the routine aseptic process. Examples include such as clearing component jams, stopping leaks, adjusting sensors, and replacing equipment components. Critical surfaces: surfaces that may come directly into contact with, or directly affect, a sterile product or its containers or closures. Critical surfaces are rendered sterile prior to the start of the manufacturing operation, and sterility is maintained throughout processing. Critical zone: a location within the aseptic processing area in which product and critical surfaces are exposed to the environment. Critical intervention: an intervention, corrective or inherent, into the critical zone. That's all for today's video on terms and definitions used in pharmaceuticals. If you found this information helpful, give us a thumbs up, subscribe to our channel, and hit the notification bell to stay updated on more job, immigration and educational content. If you have any questions or want to share your journey, leave a comment below. Until next time, keep dreaming big and reaching for your goals.